So hello everyone, my name is Kamal Kansou. I'm a researcher at INRA BIA in Nantes. Um, I'm going to present you work that we perform with colleagues from INRA and the University of Champagne-Ardenne, Reims from the unit FAR, and also Bert Bredeweg from the Informatic Institute of the University of Amsterdam. My presentation is entitled Testing Scientific Models Using Qualitative Reasoning and with an application to cellulose hydrolysis. Okay, this is the result of the research we performed recently, a couple of years ago, and which was published in a paper in Scientific Reports. So, first of all, as you might know, there is a massive production of scientific documents, scientific studies about topics in natural science, and this is represented here in this citation graph that we designed from a query in the Web of Science, a query about cellulose and hydrolysis. So this is only the first 500 hits sorted by relevance, but there are more than 4,000 publications about cellulose hydrolysis, and so obviously this is all squattered information, and the challenge for the researcher is to integrate, to digest all this information to develop an overall understanding of the phenomenon under investigation. So there is obviously a risk of information overload because each piece of scientific publication is already quite difficult to integrate, to really understand. So combining all this information is really challenging. So how can we make progress about that? So when one topic is investigated in many scientific papers, there are several explanations and theories which are proposed in these different papers, and well, different questions arise. For example, do explanations complete each other? Does the last explanation, the most recent explanation, confirm the previous ones? Do explanations contradict each other? Can a theory explain the result from other studies? Well, this last point is particularly interesting, and it was addressed in our work. So when you want to assess a theory from one publication with result from another, there is a series of limitations and constraints for the one who wants to do that. First of all, experimental records are obtained in specific, unique laboratory conditions, and it's not obvious to represent exactly this lab condition. So you will always have variability, even if it's exactly the same experiments. So numerical accuracy, it can be a limitation. Second point is that theories and results are expressed in various formats: text, diagram, mathematical model. So how do we combine all that information? We need to find the right formalism to combine all this and to integrate all this in one common representation. Third point is that theories focus generally on a specific mechanism, and they are not presented as a system or even less as models. And if you want really to implement a theory and to test this theory against result from others, from other sources, from other publication, from other experiment, then it's really an advantage to have this theory implemented as a system or as a mathematical model, even better. So classical approach to test these theories against results is to first select a bunch of papers that present theories, to capture these theories as a series of mathematical models, and to generate, to measure independent data from your laboratory. So a unique data set that you produce in your condition, and you review the list of mathematical models, physical model against this independent dataset. Okay, this approach, well, that you can find in several publications independently of the domains, have a couple limitations. First of all, the resources and data from one lab are limited. Different mathematical models can match the data, so fitting data is good but not sufficient. And finally, standard mathematical modeling formalism captures imperfectly the theory. For example, usually in publication, theories are expressed using causal effect relationships, and you cannot really capture this causal relation in, for example, ordinary differential equations. So it's important to have a representation that captures the knowledge explicitly. So our proposition is to try to capture the observation from papers about the phenomenon, so that we can really exploit the massive observation available in the literature. So how can we do that? First of all, to define a framework and a modeling approach to do that, we need to capture theories as computational representation, and this computational representation should represent as much as possible the knowledge, the scientific knowledge. So that's why we think that a causal relationship is good, is important to be captured in the model. We also want to capture various observations about the phenomenon from various sources. And finally, we want to compare and analyze the simulation versus observations. Of course, how do we do that? Keeping in mind that, of course, observations are obtained in different conditions in different contexts. So there are a bunch of modeling techniques that we can review in order to perform this testing of a scientific model. So I've listed 3 of them: conceptual mapping, ordinary differential equation, and qualitative reasoning, more precisely qualitative process theory. So qualitative reasoning is a field from artificial intelligence, and the goal of qualitative reasoning is to implement causal reasoning, and qualitative reasoning infers behavior from structure. This is called envisioning. Also, qualitative reasoning favor implementation of compositional modeling principles, so it means that you can associate model fragments, and each model fragment capture a knowledge chunk, and starting from a scenario, you can call or trigger or not different model fragments. So it's really useful if you want to work with a library of model fragments. It's very useful to switch from a theory to another using this approach. So now if we look at the characteristic of these 3 formalism, first of all, a conceptual map can capture different kind of knowledge. It's basically a semantic graph that in which we can represent many things like causal knowledge, declarative knowledge, procedural knowledge. But it's a static representation. You cannot produce simulation from that, and also it's not really a mathematically sound language, not per se. It's particularly clear representation that can be understood by domain experts. Now, if you look at ordinary differential equation, then that's the opposite. You can produce simulation. Usually, you need a little bit of numerical data, a little bit of calibration. Not always the case, but most of the time it's the case. It's a sound language, but you cannot capture any kind of knowledge, and in particular, you cannot capture causal knowledge using ordinary differential equation. Qualitative reasoning is different. It's a little bit in between these 2 formalism. In qualitative reasoning, you capture causal knowledge and also declarative knowledge, a little bit like concept map, but because it's a sound language, formal language, you can produce simulation, and you can, as it is written at the bottom of the slide, you can uh, simulate the behavior of your system over time from the structure, from description of the structure of your system. It's also a representation which is clear enough to be understandable by domain expert, so it's relatively readable. So this is an example of QR model built with GARP3, which is a qualitative reasoning workbench developed at the Informatic Institute of the University of Amsterdam. So this model is a sample of a model about enzymatic reaction, basic enzymatic reaction, in which you have the main components are the entities. There are three entities. The enzyme are called active enzyme, the substrate enzyme complex, so called here complex, and the substrate. These entities are linked together with a conceptual relation like form. Here you can see here with this relation, and in most of the case, entities are assigned a quantity, which is basically a property of this entity. For example, here concentration of substrate is a quantity about substrate. So quantity is assigned a value domain that you can see here. So value domain is composed of qualitative value, for example, landmarks like 0 or intervals like plus. Plus just here means all the value higher than 0, but it remains qualitative. There is also sign derivative, which represents the direction of change of your quantity over time. Finally, quantities of this model represent the variables of the model. They are linked together with what we call causal relations. There are two types of causal relation: proportionality, which represent the indirect influence dependence between two quantities, and the influence, which represent the direct influence between two quantities. So I will come back to this in the next slide, and I will explain this a little bit more. So from this you can also represent algebraic relation between two quantities. Here, for example, it's a product. You can representation of product between two concentration. From this, you can generate a simulation which takes the form of a state graph, and this state graph represents all the states that can be reached by the system over time. So here's a state graph of 8 states, and we have selected one pathway in red, and this pathway represents just one possible behavior of your system. So now if I look at the values of the variables in this pathway, so for example, this is an example with two concentration of enzyme over time and concentration of product over time. So for concentration of enzyme, we see that we have a decrease of the enzyme over time, and it reaches a minimum value in state 4, and then it goes up again. And for product concentration, there is an accumulation of product over time, over the reaction, and there is a maximum value in state 6, and this is the final value probably because there is no more substrate in the reaction. So in this slide, I present you how a QR model can map an ordinary differential equation or a reaction, a chemical reaction or an enzymatic reaction. So this is an example, classical example of an enzymatic reaction with E for enzyme and S for substrate and P for product. So this is represented here we can see that the first reaction is reversible here because you can produce complex enzyme substrate from enzyme plus substrate, but also the complex can is not stable and can form enzyme and substrate again. So it's not a one-way reaction here. So we can extract one differential equation from this, and this differential equation represents the evolution of the derivative of the substrate over time for a unit of time, and you can see two terms. One term is the rate of consumption of the substrate following this arrow, and you can see that it's a product between the concentration of enzyme and the concentration of substrate, and this term represents the rate of formation of the substrate over time, so this arrow, and this is dependent on the concentration of the complex enzyme substrate. So if we want to represent that in QR, so we have this kind of formalism for the rate of consumption. We say that the concentration of enzyme and the concentration times the concentration of substrate is positively related to the rate of consumption. We do the same for the rate of formation. Here, the concentration of the complex is positively related to the rate of formation. And finally, our derivative of substrate is the addition between the rate of consumption and the rate of formation, and this is represented with a direct influence. The rate of consumption is negatively influences the concentration of substrate, and the rate of formation positively influences the concentration of substrate. This is the way we can represent using P plus P proportionality and I influence this kind of relation. And it's quite easy, but there is some kind of direction between the cause and the effect that you cannot have in the differential equation. So now, to go back to the main topic, so we design a method for testing scientific model using this modeling approach, and we have separated two different kind of work. First type of work is to perform a domain knowledge analysis. So there are three steps here. First step is first of all to select the phenomenon under investigation and define a target system. Second step is to build a literature base with observational data and with theories. And the third step is the most important here, is to define a target behavior. So a target behavior is composed of different components, and each component represents a behavior that the target system should exhibit. So the target system should exhibit different kind of behavior depending of the experimentation, depending of the solicitation or the change performed by the scientist, and we want to capture this as target behavior. So once you have the target behavior, then you do the modeling work, the conceptual validation work. So this is step 4, where in the step 4 you select kernel theories and corresponding models. In step 5 you develop the QR models, and these QR models you want to test it against observation from the initial paper, from the source papers, to assess if the representation is consistent with the initial theories. This is what we call the encompassment test. Once your QR model has been validated and once you have shown that your QR model is a consistent representation of the initial theory, then you can test this QR model against the target behavior, and this is what we call the sufficiency test, because here you assess whether or not your QR model can explain your target behavior, and if it's possible explanation of the target behavior or plausible explanation of the target behavior. If it's not, if the test failed, then you can choose, and this is the last step, to augment the model. If it's, for example, if it's an insufficient coverage of the target behavior, and you can augment the model using additional theory um, and additional model, source model. So now if I go more into details about the application domain, so the first step is to select the phenomenon. Here we wanted to work about cellulose hydrolysis slowing down. This process and this phenomenon is really important for many industrial and natural bioprocesses. So, for example, it's really important for biofuel, and it's actually limiting for biofuel. We want to reduce this slowing down aspect. So, cellulose hydrolysis is monitored by a dosage, the measurement of the quantity of sugar released during the hydrolysis, and it takes the form of these kinetic curves. And here, this curve is particularly interesting because you can see that the hydrolysis rate is decreasing over time, and it's decreasing sharply. So we want to explain this phenomenon. It's really important. The system is composed of like this. It's described here. It's a cellulose fiber, and this cellulose fiber is digested by cellulobiohydrolase, which are enzymes that act processively at the surface of the cellulose fiber and digest bit by bit cellulose fiber. So there are also different kind of enzyme involved in this cellulose hydrolysis, especially in the nature. So the second step is to build a literature base. So here we have observation and we have explanatory model, and we want to have a list of papers about that. So for this work, for example, we use a set of 70 about 70 papers publication, but could be more, could be less, depending on the topic and depending on the precision of the models. For example, if you have a lot of mathematical model in the domain, you need less paper. So the third step is to define the target behavior. So this is an example from a paper from Chris Baguer. So here in this study, they monitor the hydrolysis here in the black curve, and in one experiment they decided to add fresh enzyme after 60 seconds here. And as you can notice in the red curve, there is a restart of the hydrolysis, clear restart of the hydrolysis. So we want to have a theory that can explain this restart, for example. So we captured this, for example, the restart phenomenon as a series of events, of qualitative events described here. For example, the addition of free enzyme is represented by plus here, and this triangle, the symbol which represents an increasing, the increase of free enzyme concentration. Also, at the very beginning of the simulation, the hydrolysis rate or the catalytic rate should be stable. This is represented by this symbol. And we expect that because of the restart, the rate will increase again as a result of the addition of free enzyme. And then when the addition is over, we expect that the rate will settle down. So in this step, the target behavior is about capturing salient traits of observational data, a series of events, and events must be explained by the candidate models. So in the next step, we perform the conceptual. We start the conceptual validation phase. So if you look at theories in papers, you can see that they are expressed are text, schemas, diagram, or can be mathematical model, or can be and they are also information. I would say contextual information, simulation data, corresponding observational data, goodness of it criteria, and so on and so forth. So this is represented here, different kind of representation, and you have also important result like that with a simulation curve and data, and you need to check if the fitting is okay. So these are a bunch of important information for us to collect. In the step 5, we define the baseline QR models. So this is an example selected from this publication from Moore and al. In which we have this enzymatic reaction here. And as explained before, it's relatively doable to map this kind of relation using QR model, QR formalism, and we have QR model like that. And from this, we can generate QR simulation. This is step 6. And in the QR simulation results are made of state graphs like this and value histograph like this. So value histograph represent the evolution of the variables of your system over time following a pathway, a given pathway. So it here represented in red here. So this is the evolution, for example, for the enzyme in 3 different states: free, active, inactive. So the goal here is to confront the QR simulation with simulation data from the initial publication to assert that our baseline QR model is a consistent representation of the initial scientific model or the initial theory. So this is what we've done here. We have represented the simulation curve from the initial publication, original model, and we have put the states simulated by the QR model above and checked whether or not the two simulations are consistent, or in this case, it is consistent. So we have probably a consistent mapping of the original model using QR. So here's the goal. As I say here, we test if the QR model is a correct representation of the theory. This is what we call the encompassment test. Next step is to check whether or not the QR model is consistent with the target behavior. To do that, we generate a bunch of QR simulations following different kind of scenarios. Actually, it is usually one scenario for each target behavior components. Here we have 3 target behavior components. First of them is represent the catalytic rate decreasing over time. This is represented in T3 here, time index T3, and you can see that the catalytic rate should decrease, which represents a slowing down of the cellulose hydrolysis. The second target behavior component is about the restart phenomenon. I've already presented that. And the third component is about also a restart, but with an additional limiting process that limits the restart phenomenon. And we have good evidence. We have that such a limitation process exists in the real system. So this is an example of, and here we check whether or not a target behavior can be explained with the model. And here we can see that the model can explain 2 target behavior components, but cannot explain the third one. So it was necessary to augment the model with some additional information to explain the third target behavior component. So as a conclusion, I would say that we presented a model a method about conceptual assessment of a scientific model, and we try to answer the question, can a specific theory explain observation from other studies? So the answer to this question is yes, if we focus on qualitative behaviors and silent traits first. Then we can try probably to get into details with numerical information. But first of all, we suggest to start with qualitative approach. Also, we can answer yes if scientific models are captured as knowledge representation, for example, causal model, because this way we can assess the model structure and not the mathematical formulation of the model, so which is a little bit different. And also, we check if the knowledge representation can generate sound, non-trivial, and self-explanatory simulation result, again, because we want to assess the model structure and not the mathematical formulation of the model. So we need self-explanatory simulation result. And of course, it's better if it's non-trivial. Otherwise, just an expert can do it just by reasoning on the theory. Finally, I'll say that the conditions for evaluation are a set of target behaviors. This is a concept that we have introduced here. And this concept behavior should be as diverse as possible to represent all the possible behavior of the phenomenon under investigation. So it's better if a lot of experimentation are captured in this target behavior. For example, restart phenomenon, but it could be many other experiments like that. And finally, I would say that the QR model fragments can be integrated and redeployed in different models to test putative mechanism. So I haven't presented this aspect. It's related to the concept compositional modeling aspect, and this is what we use to perform the model augmentation of a QR model and to actually change the structure of a QR model, and this way test different version of theories and different partial theories and so on and so forth. So it is also very useful for this work. So thank you for your attention.